Hi, I'm Daphne. Today I will talk about DNA metabarcoding of cloacal swabs provides insight into diets of highly migratory sharks in the Mid-Atlantic Bight. So, before I start my topic, I want to explain what is DNA metabarcoding. DNA metabarcoding is different from the original DNA barcoding. It can identify many taxa within a sample. So let's continue to introduction. At the beginning, I want to introduce the study area, Mid-Atlantic Bight, also known as MAB. And this area is near New York, and it extends from coastal Massachusetts to North Carolina. And this area also a critical habitat for migratory sharks, especially during summer. And the sharks in the north shore water of the MAB is dominated by sandbar shark, dusky shark, spinner shark, sand tiger shark, and black-tick shark. In addition, the sharks in the offshore water of the MAB is dominated by blue sharks, shark mako sharks, thresher shark, and white shark. And this species is more pelagic. Regarding seasonal migration, these 9 shark species will migrate into the MAB and they will stay here during summer and fall. So why do we need to study the diet of sharks in the MAB? Over the past century, there is a decline in sharks pollutions in this area mostly because of fishing. So we all know that sharks plays a critical role in food webs. So sharks also influence the marine ecosystem. So diet analysis of sharks is important to sharks conservation. However, the dietary habitats during the migration of these sharks still unknown. Thus, this study used DNA metabarcoding of fDNA from cloacal swab for diet analysis. So the researchers used cotton swab to collect fecal sample, and this method is minimally invasive technique. Furthermore, this study there has been proof that this way can get the same results with stomach content data. So the purpose of this study is to quantify major taxa supporting sharks biomass during the seasonal present in the MAB. Also check if the species has specific dietary preference that may show they share resources differently. So I hope I will convince you in this talk that using biotechnology to analyze diet could help shark conservation. Let's move on to the material and method. About sample collection, they caught the sharks by fishing during 11 cruises in Mid-Atlantic Bight from 2019 to 2022, and the sample size is 58. And this picture shows the sampling site of this study. After they catch the shark, they will use the cartoon swab to insert into their cloacals, and they can get the fecal DNA. Then they will do the experiments with following step in the laboratory. Here is the result. In this pie chart, it shows that there were 4 kingdoms. Four kingdoms were identified in the sequence read. And in this pie chart, we can see the number. The number means sequence reads. And the read is an inferred sequence of base pair. What is base pair? One two of the nucleotides means one base pair. Okay, so in this pie chart, we can observe that the animalia occupy the highest proportion of the sequence rate, accounts for 87.1%. And the other three kingdoms were considered to results from the contamination. So this study just focused on the animalia to discuss. As for this bar chart it shows the numbers of sample of different species. So we can see the dark blue means the list species and how many sample they catch, and the light blue means list species. How many of this species detects the fish species. So it is obvious that not every sample they detect the fish species, only 28 samples. As for this pie chart, we can show that fish dominated an animalia sequence, and the Actinopter region occupy the highest proportion of the pie chart, accounting for 85.1%. And following is elasmobranchi. Furthermore, the researcher also used two ways to know the dietary preference of the shark. The first one is the frequency of the presence of the prey. So in this table, we can show that the dark blue means this prey presents, and the light blue means this prey didn't present in the sample. So overall, the fish taxa consist of the 8 orders, 9 families, 11 fish species, and furthermore, we can observe that this row means the frequency of prey presence. So we can observe that 3 of these has the highest number, and they are Atlantic menhaden and Atlantic mercurial and little skates. Another way to know the shark's dietary preference is use prey rate frequency in different shark species. In this bar chart, we can below the chart is the abbreviation of the shark species, and in the left side of the chart is prey rate frequency. So we can see that on the left side of the line, Atlantic material and Atlantic menhaden and little skates is dormitory, and in the right side of the line, the Atlantic mercurial is dominated. So discussion. As for the dietary preference, not only the frequency of the presence of the prey, but also the prey rate frequency show that the sharks prefer to eat at least 3 species. In addition, the researcher also found the waste food web situation in this study. This food web is different from the typically food webs. It is controlled by lower traffic level specific, which are low diversity, and it is vulnerable to collapse. So if the sharks rely on the specific fish, such as this key prey in our study, so they will face the problem of food shortage. And they also found the relationship between prey and migration. In North Shore water in the summer, when the pelagic fish species increase, the acoustic tracking data of these sharks also increase. So the sharks is highly rely on this prey. So this study achieved to the proposed set previously. For the first one, they found that prey is mainly composed of raving and elasmobrain fish. Another one is they found key prey were identified, including these 3 species. Moreover, there is a strong relationship between prey and migration. In conclusion, by dietary analysis, researchers can identify the primary prey of apex predator enable to the conservation of their resources. Thank you.
As you mentioned, the MAB, Mid-Atlantic Bight, and you say that's critical habitats for migratory sharks during summer. And I want to ask that, is it only during the summer or like the spring and summer?
He asked whether they only migrate in summer. The answer is no, because in this enemy we can saw that they will migrate from the winter, and they will from the spring. So the spring and summer will near the MAB, and they will stay here summer and fall. But your question is whether it's only migrate in summer, but also fall is also there. But maybe in the fall the number of the shark is not many as in summer. Yes.
I think it's on page 16. There's a graph. I don't understand.
Okay.
Dark blue and what it means.
You mean this one and this one?
17, sorry.
17. Okay. Oh, here is the prey. Here is the prey, the species of the prey. So as for this one means the dark blue means this prey presents in this sample, and the row is different samples of the sharks. So for example, sharks A, they eat it eats this kind of prey, and shark B, it didn't eat this kind of prey. Okay, thank you.
I want to ask you, just say that on the PPT there are 28 cultures can be detected species. Does that affect the results?
She asks whether the samples on the number of the sample only 28 will affect the results. In my opinion, I think yes, because I don't know why the sample is 58, but only 28 samples detect fish, and how about another 30? So in my opinion, I think it will affect the results very, very highly. Yes.
Thank you.
Thank you.
So I want to ask about if there any difference between the fish you detect and the local fish fauna. Has this paper compared?
He wants to know about whether the sharks, the presence of the shark is related to the fish in the local fish, right? Okay. So in this slide, they found that they used the data from the past paper. They found when this two kind of species is increasing, then the sharks is also increase. So, but only in summer.
So these two species of fish is the dominant fishes in that area.
I think no, because they are only increase their presence only in summer. So I think that they will also migrate. Yes.
And I think only 11 fish species, very few.
Yes. My
Thank
data
you.
is more than 11.
Thank you.
Thank you.
Yeah.
Okay.
So I would like to know, like, how does the sharks being caught? Like, do they have like snake cages? And then like after you catch a sharks, right? Because they're sharks, like how do you insert the cotton swabs and get the FDNA? I think this is like very a tough
Hard,
thing to do. Yeah.
Thank you for your question. So to repeat her question, she wants to know how they catch the shark and how they do the experiments. Okay, because my experiment is the same. The step is the same as this paper. So in this paper, they will go to the sea by the boat, and they will use the long line to catch the shark. Then after they catch the shark, they will turn the sharks, use this way to up the face the sky. Then they will separate the pectoral fin and use the how to say use something to swab the seawater. Then they can use the cloacal swab to take the sample. Okay. Thank you.
I think Daphne gave a very nice talk. So her talk speed was smooth and her slides were well prepared. Wherever I thought, I don't know that turn, then she immediately explained the turn with the image that was very useful. But still there are, so let's talk about her good part. So you can see that her slides were full of artistic design. You might think, well, I can also do that, but then you have to do that. Then this will give your slide a sense of style, okay? And then she also used the advanced skill that I introduced in my lecture. So where you can say, I want to convince you, okay, what is the main message that I want to convince you? That's also the advanced skill that she used. And then she always, I like this kind of, that was also very nicely made. And then I think that was a self-made figure, is that right? So you can see that you, as a student, can also make very professional scientific graphics using Excel. And then she has this, you see these are so super small, and she basically used this just to show the pattern of the data, but at the end, she summarized the data with a nice text box with large font friendly to me. And then she also always add image of the fish so that I know, oh, well, okay, that is the little skate. Otherwise, I really have no idea what is little skate. So these are all very friendly to the audience. And then also during her discussion was also easy to follow. Where did you start the discussion? Okay, like here, as I introduced in my previous lecture, that it's always easy to show the flow of logic by using arrows. And here she used these arrows, so it's very easy to understand. And she always remembered to add this little smug shot of the fish or the shark, so that's also very nice to me. Although even after looking at this figure, I still have no feeling for them, but still that looks really nice, no? Compared to slides without smug shots. And then I also like her conclusion slide, so you can see that she reused her purpose slide. So that's a skill that you should definitely learn, that you can reuse all of your purpose slide to make sure your conclusion is matching your purpose. But next time I would suggest that maybe you don't need to hide the purpose, because I really want to see how your answer corresponds to your purpose. So when you do this, I cannot check. Okay, so you can simply display it here, here, okay? Just display your answer here, okay? Don't hide your purpose. Then you can change these two conclusions. Okay, then some minor things to answer. So I think for students who present DNA metabarcoding, because every year there are students presenting that, I think you probably need to explain the advantage of this technique, especially you say it's a non-invasive or minimally invasive technique, then you need to explain in comparison what is the traditional technique, okay? Does that involve killing a shark, for example? And then also, I think you really need to explain, what is that, okay? Cloacal swabs. I really need to look into the dictionary. And then there are also some, from time to time, some small terms that still needs explanation. For example, like here, like you always add image. However, I really have no feeling what's that. So please use some simple explanation or image. And then, yeah. And then, okay, I think this figure, I got a little bit confused at some point. So maybe you can explain what are these by printing out. What are these? Like, is it abbreviation of sharks?
Yes,
This?
I mentioned it.
Yeah, you mentioned it but you are constantly talking, and then you put the shark. But these are not shark species, right?
Yes.
Okay, so I was really getting confused. So, okay, so what are these? That? This? That? So I really get confused. Okay, so maybe you can move them somewhere else in order not to confuse me. I was really thinking, is this a shark? Okay. Okay, but otherwise I think it's a very nice talk, so let's thank Daphne.